Hello friends, welcome to my channel Navid Biochemistry Classes. Thank you for your support. In this video, we will talk hydrolysis of protein, chemical hydrolysis of protein. Okay, when you talk chemical hydrolysis, obviously you're talking chemical substance excluding enzyme. So chemical hydrolysis we have already discussed in our previous videos. It can be acid catalyzed or it can be base catalyzed or it can be enzyme catalyzed. But here we will talk only right now chemical hydrolysis, that is hydrolysis of protein either by acid or by base. So when you talk protein, obviously you know protein is a complex biomolecule and it is polymer of amino acid as a monomer. And when you talk amino acid, obviously as the name suggests, it is a compound which contains both amino group and a carboxylic group and represented by general formula base. So here you have NH2CHR pseudoloid amino acid. Formula is there. When we say protein is a complex compound, it has three layers of a structure, that is primary, secondary, tertiary and quaternary structure. That is not interested, we are not interested in this. But I would like to give some input about the amino acid. Well, amino acid, as I told you, is a substance which have an amino group and a carboxylic group. So when you say protein, you are saying that it is a biopolymer or biomolecule. So all naturally occurring amino acid except glycine are optically active. So what is glycine? You can see the structure is glycine is NH2CH2 pseudoloid, so no chiral atom, so no optical activity. So other than that, in general, all amino acids are optically active. They can be dextrorotatory, okay, and they can be liberatory. That is plus and minus sign you will find. But essentially all amino acids exist as L isomer. Okay, L isomer, that is we are talking L configuration, that is representation of the isomer in terms of DNL. So here we will confine only that amino acid, naturally occurring amino acid has L configuration or it exists as L isomer. So what will be the representation of L isomer? You see, well, this will be the representation of L isomer. This represent L isomer of amino acid. Now you can see here NH2 is on the left side. So in L isomer, when you write like this, okay? That is, so it means that is we are writing the amino acid in terms of L configuration. And obviously, if you here you have NH3, and this is deuterium form of the amino acid. I think as a beginner you must be knowing all. In coming videos we will explain all this, but let us now confine it to this amino acid fundamentals. Now we said that L isomer is there but in some cases D isomer also exists. D isomer of amino acid also exist. Where? Exist in well, it exists in peptidoglycan of bacteria cell, and some exotic sea dwelling creatures, sea dwelling animals or creatures, what you want to say. So it is not that it is final, but we are talking only D isomer of amino acid you will find in some cases of exotic sea dwelling animals, and of course in peptidoglycan of bacteria cell wall. In bacteria cell wall. So as we discussed that protein are polymer of amino acid. So when two amino acid combined, they form peptide. Okay, and they combine to form, and further tripeptide. Peptide is also called dipeptide. If three combine, that is tripeptide. And of course you have so many amino acid combining with each other. You call it as a polypeptide. Now when two amino acid combine and they combine it, they are linked to each other by CONH bond, and you call it peptide linkage or bond. Okay, and obviously this is amide. You can see it. This is amide bond. The functional group, you can see it. Amide bond is there, and obviously this is a polypeptide. This is a polypeptide, and obviously this particular you will call, that is two amino acid. You see here, if you have two amino acids, CHR1 pseudo-OH, and you have NH2CH R2COH, what will happen? You will get this condensed product. This condensed product, it's like this in a simpler form. Okay, so obviously this is your peptide linkage. You call it peptide or dipeptide, and obviously it goes on. Now what is the striking here? You can see always when you write a peptide, you keep NH2 left. Well, this is, you say that this is N-terminal. That is, amino is kept left while writing the structure of polypeptide. You keep NH2 left, and obviously the carboxylic group is at the right, and you call it C-terminal. C-terminal, and obviously you are keeping on in right, or rather at right. Okay. Now sometimes you will find you just represent it by OH, and you represent the NH terminal by H. You will also find it like this, that is H and OH at the right end. Okay, so this you represent, and obviously the polypeptide, as I told you, that when you talk protein, you are talking four different layer of a structure, that is primary, secondary, tertiary and quaternary. And this polypeptide and linear structure of protein, you can also call it as a primary structure of protein. Okay, so this is the primary structure of protein we are talking. We are not talking some complex. Well, after a brief summary, or rather introduction of amino acid, let us talk the topic acid hydrolysis of protein. Now, acid hydrolysis of protein is achieved by refluxing protein with acid, that is, it can be aqueous HCl or H2SO4. And what will be the result? Actually, the acid will break the peptide bond. Obviously, the monomers of protein are attached with peptide bond to form the polypeptide and hence protein. But if the hydrolysis is breaking the peptide bond, so what you will get? You will get a mixture of amino acid. Now you can see that if you have this polypeptide and you do hydrolysis, with water and acid catalysis. Obviously, you have acid catalysis hydrolysis. You can see we are talking only this, that is, it is not reversible, it is irreversible. And so what you will getting, you see here, the H of water will come here, and similarly here you have HOH. So what will be the result? Result, you can see that you will be getting a mixture of individual amino acid. So you have amino acid is there. You are getting 3 amino acid. As there are 3 components, you will get the corresponding amino acid mixture. So acid hydrolysis of protein results into formation of amino acid. Now one thing is interesting, if the protein has tryptophan, now tryptophan, well, tryptophan is an amino acid. If present in protein, undergoing acid hydrolysis, what will happen? Tryptophan will be destroyed. Tryptophan will be destroyed. So what is tryptophan? You can see tryptophan is here. All this is into derivative. See here. CS2. Okay. H is there. So this is your tryptophan. So what I want to convey, that if you have a protein which contains tryptophan, you will be not getting on hydrolysis tryptophan, it will be destroyed. And apart from that, you can see we talked that optical active amino acids are optical active. Now in acidic hydrolysis of protein, another feature of acid hydrolysis of protein is that racemization of amino acid does not take place. That is, generally amino acid optical activity is intact except for tryptophan, that is destroyed. Okay, and the last one is that in some protein, when acid hydrolysis is carried out, what happens? A black substance is produced which is insoluble in acid, and that is called humin. Now you see here, during acid hydrolysis of protein, no racemization of amino takes place. Racemization is the process of converting an optically active compound into its racemic mixture. So here racemization is not taking place, so optical activity of amino acid is retained. And the last one, I told you that in some cases a black substance is formed which is called humin, and this is a carbon based macromolecule. So this is all about acid hydrolysis of protein. Thank you.